For most of history, the effects of meditation were described in experimental and spiritual terms, impossible to verify, easy to dismiss. Then neuroscience happened. Holzel et al.'s 2011 psychiatry research paper showed that 8 weeks of mindfulness meditation produced measurable increases in gray matter density in the hippocampus, posterior cingulate cortex, and cerebellum, while producing gray matter reduction in the amygdala, the brain's threat response center. These are structural brain changes, observable on MRI, produced by a daily 45-minute practice in under 2 months. Holzel et al.'s finding that amygdala gray matter density decreased, while hippocampal density increased, after 8 weeks of MBSR represents one of the clearest demonstrations that meditative practice physically restructures the brain's emotional regulation architecture. The amygdala reduction correlates directly with self-reported stress reduction and is accompanied by reduced salivary cortisol in the same participants. This video draws from 5 peer-reviewed studies across psychiatry research, PNAS, JAMA Internal Medicine, and Nature Reviews Neuroscience, covering structural brain changes from meditation, gamma wave synchrony, inflammatory gene expression, clinical meta-analysis, and the broader neuroscience of mindfulness. Meditation has historically been treated by Western medicine as a complementary, unverifiable practice, adjacent to wellness culture and without serious clinical standing. Goyle et al.'s 2014 JAMA Internal Medicine meta-analysis, reviewing 47 randomized controlled trials covering 3,515 participants, changed this assessment. The review found moderate evidence that mindfulness meditation reduces anxiety, depression, and pain, with effect sizes comparable to antidepressant medication without the side effect profile. Creswell et al.'s 2014 PNAS study added a molecular dimension to the meditation-health relationship. 8 weeks of MBSR training in lonely older adults not only reduced subjective loneliness but produced measurable reductions in NFB-driven pro-inflammatory gene expression, including reductions in CXCL10, Illinois-8, and TNF at the gene expression level. Meditation produced changes in immune gene activity that are clinically relevant to aging and disease. Meditation's effects span multiple neurological, psychological, and physiological domains. Tang et al.'s Nature Reviews Neuroscience synthesis of the neuroscience literature documented changes in cortical thickness, white matter connectivity, default mode network activity, and subcortical structure, making meditation one of the most neurologically well-characterized behavioral interventions. Holzel et al.'s 2011 MRI study represents the strongest structural evidence for meditation-induced neuroplasticity. The study used voxel-based morphometry to precisely measure gray matter density before and after 8 weeks of MBSR in 16 participants compared to 17 controls, with blinded image analysis. Lutz et al.'s PNAS study on long-term meditators used EEG to document a phenomenon not seen in non-meditators: high-amplitude gamma frequency, 25 to 70 Hz, synchrony sustained across widespread cortical areas during compassion meditation. Gamma synchrony is associated with attentional binding, conscious perception, and high-order cognitive processing, and long-term practitioners produced it voluntarily. Meditation produces measurable improvements across multiple health domains, each validated across multiple studies. The evidence base allows a comparison of the strongest documented effects by magnitude and consistency. Meditation produces its neurological and physiological effects through a cascade of changes in neural circuits, neurotransmitter systems, and autonomic regulation. Understanding this mechanism explains why different meditation types, focused attention versus open monitoring versus loving kindness, produce different patterns of change. The evidence-based meditation framework selects the meditation modality best suited to the intended outcome: structural brain changes, anxiety reduction, inflammatory gene expression, or long-term contemplative depth, and builds a sustainable practice protocol. The timeline of meditation's neurological and physiological effects progresses from immediate autonomic changes to structural brain modifications over months. Implementing a daily meditation practice requires 6 consistent actions that build the habit, select the right modality, and progressively develop the attentional skills that produce documented neurological change. Meditation practice should be personalized based on the primary goal: anxiety reduction, cognitive performance, pain management, or structural neurological change. Different modalities are optimal for different outcomes. Meditation is one of the most neurologically well-characterized behavioral interventions, with effects spanning from gene expression to brain structure. Here is what the science shows. The evidence is clear. The mechanisms are understood. And the neurological changes from meditation are within reach for anyone who establishes a consistent daily practice. Follow MedBrief Today for more science-backed health insights, explained clearly.